Hi, and welcome to Miss Elliott's Biology. Today we're going to be talking about the cell cycle and how a cell divides. All right, so a cell's life really involves really three kind of main parts to it. The first part is interphase. So a cell is born and it goes through interphase where it basically does its job and it also grows. The second part is, these two actually parts are when a cell divides. So the two parts of cell division are when the DNA divides and then when the rest of the cell divides, the cytoplasm, all the organelles, and the membranes, right? So before we get started, I just want to show you the different examples of how cells go through their life cycle. So brain cells actually are all developed during our childhood, and they don't grow and they don't divide after that. So really, once they are created, once they are born, cells will start to live and start to do their job and go into what is known as the GO cycle. All right, and that's just when the cell cycle is arrested. It does its job, but it doesn't grow and it doesn't divide. Now, hair follicle cells are very different. So our hair follicle cells are at the very base of our hair, and we know that hair grows pretty quickly. We got to get it cut all the time. And so these cells right here in the follicle are going to divide rapidly, and they are going to make new hair cells. And so they, in contrast, are going to go through this life cycle very quickly and make more and more and more cells. So just a difference between two different types of cells. So the first part of the cell cycle, as I mentioned before, is interphase, the cell living its life. So G1 is the first phase of interphase, and really all that's going on there is growth. So growth of the cell, making new organelles, and doing its job. The next part of interphase is the S phase, and S stands for synthesis. And synthesis is just making DNA. So in another podcast that I have, I talk about replicating DNA, and you can look at that to see kind of more about this process. But right now I just want to do an overview. Really what we're doing during the S phase is DNA replication, when you go from one chromosome and you duplicate it and you get one chromosome that is duplicated. So this really defies all mathematics, but really we go from one chromosome to one chromosome with two copies. And the reason we still consider it one chromosome is because this centromere right here, so I'm going to actually do this in red so you can see it a little bit better. So this centromere, I like to call it a belt. It holds the two parts together, the two copies together. And each copy is called a sister chromatid. It is exactly the same. It's just copied, a copy of one another. And these two are held together by the centromere. And that's what we're going to divide later during DNA division of mitosis. All right, so the second part of a second growth phase of interphase is called G2. It's after S, so we know that now we have two copies of every chromosome, and we're going to continue to grow the cell. So the cell is going to continue to grow. We're going to get more organelles, and really now in this G2 phase, we're going to prepare this cell for cell division. So we're going to make sure it's big enough and it has enough organelles so that we can go into mitosis. All right, so the first part of cell division, as I mentioned before, right here, is mitosis, and it is the division of the DNA. So just nuclear division is what we call it. So as kind of a reminder, I want to show you that this is interphase. And interphase comes before, and what's really important about interphase is that the DNA is relaxed. It's relaxed, it's being read, and so it actually gets this kind of noodly form that you might not recognize. This noodle form is called chromatin, all right? And chromatin, what that looks like in a cell is it just kind of looks like a mass. You can't really tell the difference between any of the chromosomes here because it's so relaxed you can't really see it. Now, once we get into mitosis, the first part of mitosis is when you're going to actually see the DNA condense really tightly into this X-looking chromosome. Very important. All right, so we've officially moved into mitosis by condensing the DNA. We've changed it from chromatin into a chromosome form. Now, this part where it starts to condense is called prophase. Prophase is the first step of mitosis. All right, so in mitosis, you also have the nucleus starts to break down, and that's really important because the DNA needs to be able to move all over the place, actually move to the poles of the different poles of the cell so that we can divide it. We also have here centrioles. Centrioles are basically tow trucks. They help to pull the DNA to opposite sides, and so do spindle fibers, which are basically the ropes of the tow truck. All right? So during metaphase, this is our second phase of mitosis. So prophase is the first, metaphase is the second. And metaphase is conveniently starts with an M, so we can remember that by it lays in the middle. So all the DNA, all the chromosomes will line up in a single file line in the middle of the cell. We also call it the metaphase plate, which is at the middle plate, or the equator. So now we can also see that the spindle fibers have actually attached to both sides of that centromere, that center belt that we had in the middle of our chromosome. So the next step is anaphase, and anaphase is important because we actually start to see the DNA divide. So here we have one sister chromatid being pulled one way, and then we have our other sister chromatid being pulled the other way. All right, so half of the DNA is going to one side and half the DNA is going to the other side. And finally, what we get in telophase is we get two new nuclei that are starting to form that are exactly identical to one another. The DNA starts to relax back into its noodly chromatin form. All right, and these chromosomes, you have one of each, just like you had one of each to start with. So we're really getting exactly the same cells that we started with, and two of them. All right, so real quickly, just look at these images. You can see the difference here. You can't see the chromosomes because DNA is in the noodly chromatin form. Here you can see individual chromosomes. Here they're all lined up in the middle. Here they're being pulled apart. And here you see two nuclei starting to redevelop. The next part of cell division is cytokinesis, and this is a very, very simplified image of that, where the cytoplasm and the organelles are divided. So really we just need to, we've already got the DNA separated, we need to divide the rest of the cell. Now it's a little bit different between animals and plants, so let me just show you that real quick. So animals, animal cells do not have cell walls. So really conveniently, we don't have cell walls, and we can just form this contractile ring and pull from either side. And as we kind of pull in at either side, we just pinch off the two new cells. So we don't have to do much, we just have to really get that in between those two nuclei, we have to pinch it off to form two new daughter cells. Plants, because they have a cell wall, what they do is they start to form cellulose in between the two nuclei. All right, this forms a cell plate, and then eventually that cell plate will form a new cell wall between the two nuclei. All right, so this is just, again, another kind of image. I'll go through this quickly with you. Here we have interphase. You can see the DNA is all in the chromatin form, so you can't really distinguish. Here you have prophase. You can start to see the chromosomes form. Here you have metaphase, where all of the chromosomes are lined up in the middle. Here you have anaphase, where you start to see division between the chromosomes. In telophase, you start to see two new nuclei develop. And then finally, in cytokinesis, you start to see a cell wall or a cell membrane form between the two, and then each will go into its own interphase part of the life cycle. All right, thanks for watching.